Welcome back to the deep dive. Today we are really cracking open the playbook of some of the planet's most successful invaders, pathogens. And we're starting with something maybe unexpected, a Nobel Prize discovery about, well, cellular housekeeping.
That's right. We're talking about the 2016 Nobel work on autophagy. Basically, it's cellular self-eating. It sounds a bit dramatic, but it's the cell's internal recycling program.
Okay, recycling, like getting rid of old parts.
Exactly. Old organelles, damaged proteins, recycling nutrients when the cell is starved, and crucially for our topic, it's a defense mechanism against intracellular pathogens. It cleans them out.
So it's an internal cleanup crew, different from phagocytosis, right, where immune cells kind of gobble up stuff from the outside.
Precisely. Autophagy is internal housekeeping.
Yeah.
But here's the kicker, the sort of counterintuitive part. The really nasty, successful pathogens, they actively fight this cleanup process.
They sabotage the cell's recycling system. Why?
Well, think about it. If the cell can't clean itself up, the pathogen has free rein. We see this with bugs like Salmonella entericus, Serovar typhimurium, and Mycobacterium tuberculosis, the TB germ.
So they have specific tools like molecular weapons just for this, these virulence factors we hear about.
Absolutely. Salmonella, for instance, makes proteins that specifically degrade the host proteins needed for autophagy to even start. It's like cutting the power cord on the garbage disposal.
Wow.
And this lets the pathogen hoard nutrients inside the cell and just replicate like crazy. Even viruses do it. Herpes simplex virus type 1, HSV1, it is a protein that blocks a key autophagy player in nerve cells.
And if it can't block it?
Well, researchers found that mutant viruses without that blocking ability couldn't cause lethal encephalitis. It directly shows how vital suppressing this cleanup is for the pathogen to actually cause severe disease.
That's incredible. Okay, so that sets the stage. Our mission today is to walk through this whole process, defining these infectious agents, how they get in, the amazing ways they dodge our defenses, and ultimately how they cause harm.
Sounds good. So let's start with some basics just to make sure we're all on the same page. First off, infection versus infectious disease.
Right. An infection is just the microbe being there, growing on you or inside you.
Yes. But an infectious disease is when that presence actually makes you sick, impairs your normal functions. The microbe is winning, essentially.
And how do we measure how bad they are? We have those two terms.
Pathogenicity and virulence. Pathogenicity is simply the ability to cause disease. Does it have the potential? Virulence is the degree of harm. How nasty is it? A highly virulent pathogen causes really severe disease.
Okay. And what about those microbes that are usually fine, but sometimes turn bad?
The opportunistic pathogens. These are often part of your normal microbiota, the bacteria living harmlessly in your gut, say. But if your immune system gets weak, or if they end up somewhere they shouldn't, like E. coli from the gut getting into the urinary tract.
Then they cause trouble.
Exactly. They seize the opportunity.
Makes sense. Now, where they live in the host also matters, right? Their habitat.
Absolutely. It dictates their strategy. You've got the extracellular pathogens. They stay outside host cells, in tissues, blood, fluids. Think Yersinia pestis, the plague bacterium. It hangs out between cells.
Okay, so they don't invade cells themselves.
Correct. Then you have the intracellular pathogens. These guys actually live inside our cells. Big advantage there, hiding from a lot of the immune system.
And there are different types of those too.
Yep. Facultative intracellular pathogens are flexible. They can grow inside cells, like Brucella abortus, but you can also grow them in a lab dish, in pure culture.
But some have to be inside.
Those are the obligate intracellular pathogens. They absolutely require a living host cell to replicate. All viruses fall into this category, obviously. But also some bacteria, like chlamydia. They basically outsourced all their metabolic needs to the host cell.
Okay, so we know what they are, where they live. What about the timeline when someone gets sick? Does it follow a pattern?
It generally does. There's a typical course of infectious disease. First, after the pathogen gets in, there's the incubation period.
That's when nothing seems to be happening.
Right. The pathogen is there, it's multiplying, but there just aren't enough of them yet, or they haven't produced enough toxins to cause noticeable symptoms. It can be hours, days, even years.
Then what?
Then comes the prodromal stage. This is often tricky. You feel off. Vague symptoms, maybe tired, a bit achy. Nothing specific.
But crucially...
You're often highly contagious at this point. The pathogen numbers are really ramping up.
Okay, that's important for spread. Then comes the worst part.
The illness period. This is when the disease is most severe. You have the characteristic signs and symptoms for that specific illness. Your immune system is usually fully engaged by now.
And hopefully things start getting better after that.
Yes. The period of decline, Signs and symptoms begin to fade, your immune system is winning, or maybe treatment is working, followed by convalescence, the recovery period where you regain strength.
You mentioned signs and symptoms. Is there a difference?
There is. Signs are objective things someone else can observe or measure. A fever, a rash, high blood pressure. Symptoms are subjective, what the patient feels. Pain, nausea, fatigue. You can't directly measure someone else's pain.
Got it. Okay, so how do these pathogens even reach us in the first place? We need to talk about sources and reservoirs.
Right. A reservoir is the natural environment where the pathogen normally lives and multiplies. It could be other animals, soil, water. Think birds for West Nile virus. That's the reservoir.
And the source?
Source is the immediate place or object from which you actually get infected. Sometimes the source and reservoir are the same, like getting rabies directly from a rabid animal, but sometimes they're different. Like the reservoir for a foodborne illness might be cattle, but the source for you is the contaminated hamburger you ate.
And when diseases jump from animals to humans, that's a zoonosis.
Exactly, like influenza, rabies, Lyme disease. Many important human diseases are zoonoses.
Okay, so how does the transmission actually happen? What are the routes?
Several main ways. Airborne is a big one, but we need to distinguish here. Large droplets from a cough or sneeze are heavy, fall quickly, and usually require close contact. Direct transmission.
Like standing too close to someone who's sick.
Right, but then you have droplet nuclei. These are much smaller, dried-out particles. They can stay suspended in the air for hours, travel long distances on air currents. That's indirect transmission. Dust particles can also carry pathogens this way.
Okay, so distance matters, and particle size. What about touching things?
That's contact transmission. Direct contact is person-person kissing, touching, sexual contact. Indirect contact involves an intermediate object, a fomite.
A fomite.
Yeah, any inanimate object that can transfer the pathogen. Doorknobs, bedding, toys, surgical instruments. If they're contaminated, they can be a fomite.
So fomites are like vehicles.
They can be. The term vehicle transmission often refers to inanimate materials like contaminated food, water, even drugs that transmit pathogens to many people from a single contaminated source. Think of a food poisoning outbreak from contaminated lettuce.
Right. And then there are living transmitters.
Vector-borne transmission.
Yeah.
Usually, arthropods, mosquitoes, ticks, fleas, they carry the pathogen from one host to another. Malaria via mosquitoes, Lyme disease via ticks.
And you mentioned something interesting earlier. These vector-borne pathogens are often really virulent in humans.
Yes, it's a fascinating point. Because the pathogen is adapted to survive in the vector, the mosquito or tick, without necessarily harming it, its virulence in the secondary host, like us, isn't constrained by needing to keep us alive for transmission. The vector does the transmitting.
That makes a chilling kind of sense. It doesn't need us to be mobile.
Exactly. We should also mention vertical transmission. That's mother to unborn child, resulting in a congenital infection. And a quick historical nod here.
Ah, yes, Semmelweis.
Ignaz Semmelweis, mid-1800s. He figured out that doctors going from autopsies straight to delivering babies were transmitting something causing puerperal fever or childbed fever. His insistence on hand washing drastically cut deaths. A tragic story, as his ideas were resisted, but fundamental proof of direct contact transmission.
A reminder of how basic hygiene is so critical. Okay, so if transmission happens, how do scientists actually measure the infectiousness or danger level?
We need ways to quantify virulence. Two key measures are used, especially in lab settings. The first is infectious dose 50, or ID50.
ID50, 50%.
Right. It's the number of microbes, bacteria, viruses, whatever needed to cause infection or disease in 50% of the animals tested in an experiment. For example, maybe it takes, say, 100,000 salmonella bacteria given orally to make half the mice sick. So the IG50 is 100,000.
Okay, and the other measure?
Lethal dose 50, or LD50. Same idea, but it's the dose required to kill 50% of the experimental group.
So wait, if a pathogen has a low ID50 or LD50, that means it's more dangerous because it takes fewer organisms to cause infection or death.
Exactly. That's the key takeaway. Lower number, higher virulence or potency. It seems counterintuitive at first, but it makes sense when you think about it as the minimum dose for effect.
Right, takes less to do the damage. Okay, so assuming the pathogen reaches us and it's virulent enough, how does it actually start the process? It has to stick, right?
Adhesion is step one. They have to attach to host cells, usually at a specific portal of entry, like the respiratory tract lining or the gut. And this isn't random. There's incredible specificity, or tropism.
Meaning they only infect certain cell types.
Yes. It's like a lock and key. The microbe has molecules on its surface called adhesins. These could be on pili, which are like little hairs, or in the capsule, or maybe viral spikes. These adhesins have to bind to specific receptor molecules on the host cell surface.
So if the host cell doesn't have the right receptor, the microbe can't stick.
Generally, no. It's a great example of coevolution. The binding is often a two-step process. First, a sort of loose reversible docking, maybe based on general physical forces. Then, a much more specific, tight, permanent binding between the adhesin and receptor that locks it in place for colonization.
Okay, they're stuck. Now what? Invasion.
Invasion is often next, especially for intracellular pathogens. This can happen actively or passively. Active penetration means the pathogen produces substances, like enzymes, that degrade the host tissues, the stuff between cells, or the cell surface itself. They basically digest their way in.
And passive.
Passive penetration is simpler. The microbe just takes advantage of existing openings, a small cut, an insect bite, maybe damage caused by another infection. It slips in through a breach in the defenses.
And once inside, some have really clever ways to move around. You mentioned listeria.
Ah, yes. This is amazing. Some intracellular bacteria, like Listeria monocytogenes, actually hijack the host cell's own machinery, specifically the actin cytoskeleton.
The cell's internal scaffolding.
Exactly.
Hmm?
They trigger actin proteins to polymerize behind them, forming an actin tail. It's like building their own little rocket engine inside the cell, using the cell's parts.
No way. And they use this tail to out.
To propel themselves through the cytoplasm and even push directly into adjacent host cells. They spread cell to cell without ever going outside, completely avoiding antibodies and other external immune defenses. Just brilliant in a terrifying way.
Absolutely terrifying. Does this kind of spread lead to wider infection?
Often, yes. Penetration and local spread can eventually lead the pathogen into the lymphatic system or the bloodstream. Having bacteria in the blood is bacteremia. If those bacteria are multiplying and causing a systemic illness, that's septicemia or sepsis. Very dangerous.
So survival is key. They have to deal with our defenses, but also other microbes, like our normal gut bacteria.
Good point. They face competition. Some pathogens have evolved ways to fight off the resident microbiota. A fascinating example is the type V's secretion system, or T6SS.
Another weapon system.
Pretty much. Think of it as a molecular spear gun. Certain gram-negative bacteria use the T6SS to inject toxic proteins directly into neighboring bacterial cells, their competitors. It's microbial warfare to clear out the locals and establish a foothold.
Wow. Okay, so they fight other microbes. How do they fight us, our immune system?
Many strategies. Immune evasion is critical for pathogens. Some produce decoys. Hepatitis B virus, for instance, releases huge amounts of a surface protein that isn't infectious, but it soaks up antibodies like a sponge, protecting the actual virus particles.
Clever. What else?
Antigenic variation. This is changing their surface appearance, so our antibodies no longer recognize them. Neisseria gonorrhea, the cause of gonorrhea, is a master of this. It constantly switches the type of those adhesion hairs on its surface through genetic shuffling. Your immune system makes antibodies to one type, and boom, the bacteria switch to another.
Always one step ahead.
Or they use camouflage. Some bacteria, like Streptococcus pyogenes, have capsules made of hyaluronic acid, which is also found in our own connective tissue. It's like wearing a cloak of invisibility made from host material. Others have capsules that prevent immune proteins, like complement, from sticking.
So hiding, changing appearance. What about teamwork? Do they work together?
Absolutely. That brings us to biofilms. These aren't just random clumps of bacteria. A biofilm is an organized, structured community of microbes stuck to a surface and encased in a slimy matrix they produce themselves. Think dental plaque, or the stuff that gums up medical implants.
And this is different from when they're just swimming around freely?
Very different. The free-swimming state is called planktonic. Bacteria in a biofilm behave differently. They turn on different genes, communicate with each other chemically, quorum sensing, and that matrix provides amazing protection.
Protection from what?
Antibiotics, disinfectants, and immune cells. Antibiotics often can't penetrate the matrix well, and the bacteria inside might be metabolically slower, making them less susceptible. Immune cells, like phagocytes, literally can't engulf the bacteria stuck in that goo.
So the immune cells get frustrated.
Exactly. It's called frustrated phagocytosis. The phagocyte tries to attack the biofilm, can't get the bacteria, and ends up releasing its toxic antimicrobial chemicals outside itself,
Which damages.
which damages the surrounding host tissue, while the bacteria in the biofilm remain relatively safe. It's a major problem in chronic infections.
Okay, this is getting complex. All these weapons and strategies, where do they come from genetically?
Often, yes. Many of the genes encoding these virulence factors, toxins, adhesins, invasion systems, the T6SS, are clustered together on the bacterial chromosome in specific regions called pathogenicity islands. or PAIs.
Islands. Like separate chunks of DNA?
Sort of. They often look different from the rest of the chromosome, suggesting they were acquired horizontally transferred from other bacteria, perhaps via viruses or conjugation, rather than inherited vertically. It's like a non-pathogenic bacterium suddenly received a badness software update. The toxic shock syndrome toxin gene in Staph aureus is often found on a PAI.
A massive evolutionary leap. So the ultimate damage often comes down to toxins, right?
Toxins are major players in causing disease symptoms. We broadly group bacterial toxins into exotoxins and endotoxins. Exotoxins are proteins, usually heat-sensitive, that are actively secreted by living bacteria. Some are incredibly potent. Botulinum toxin is one of the deadliest substances known.
And they work in different ways.
Many different ways. A classic type is the AB toxin. It has two parts. The B subunit is for biface. Binding it attaches the toxin to the specific target host cell. Then the A subunit, the active part, gets inside the cell and messes things up, often by acting as an enzyme. Diphtheria toxin, for example, uses its A subunit to shut down protein synthesis.
Okay, so B binds AX. What else?
You have membrane-disrupting toxins. These basically punch holes in the host cell membrane or destabilize it. This causes the cell's contents to leak out, and water rushes in, making the cell swell and burst, like popping a water balloon.
Nasty. And then there were superantigens. That sounds bad.
They are very bad. Superantigens short-circuit the immune system in a dangerous way. Normally, an antigen-presenting cell shows a specific piece of antigen to a specific T cell. Superantigens act like a clamp, non-specifically locking antigen-presenting cells directly to T cells, regardless of what antigen is present.
So they activate loads of T cells at once.
Exactly. A huge number of T cells get activated inappropriately, leading to a massive release of signaling molecules called cytokines. This is the infamous cytokine storm.
And that causes
Systemic inflammation, fever, leaky blood vessels, a drastic drop in blood pressure, shock, and potentially widespread organ failure. Toxic shock syndrome is a classic example caused by superantigens.
Okay, that covers exotoxins. What about the other one, endotoxins?
Right, endotoxin is different. It's not a secreted protein. It is part of the outer membrane of gram-negative bacteria specifically, the lipopolysaccharide or LPS molecule.
So it's part of the bacterium itself.
Yes. It's generally only released in large amounts when the bacterium dies and leases, breaking apart. The toxic part of LPS is a fatty acid component called Lipid A. It's heat stable, unlike most protein exotoxins.
And how does Lipid A cause damage? Is it like an enzyme?
No, it causes damage indirectly. Lipid A doesn't directly attack host cells. Instead, it powerfully stimulates immune cells, especially macrophages and endothelial cells lining blood vessels,
Stimulates them to do what?
to release massive amounts of those same inflammatory mediators. We talked about cytokines like TNF-alpha and interleukin-1. It basically triggers a system-wide alarm.
So it's our own immune response overreacting to Lipid A that causes the problem.
Precisely. This leads to the septic shock cascade, widespread vasodilation, plummeting blood pressure, shock, fever. Plus, it activates the clotting system inappropriately, leading to widespread small clots in organs, disseminated intravascular coagulation or DIC, which paradoxically can also lead to bleeding as clotting factors get used up. It culminates in multiple organ failure.
A truly devastating chain reaction triggered by a piece of dead bacteria.
Absolutely. And finally, we should briefly mention mycotoxins. These are from bacteria, but from fungi. Things like aflatoxins produced by Aspergillus mold on peanuts or corn, which are potent liver carcinogens, or ergot alkaloids from Claviceps fungus on rye, which includes precursors to LSD.
Wow. So fungi have their own chemical weapons too. Okay, we've covered a huge amount of ground here. From the basic definitions of pathogenicity and virulence, how we measure them with ID50,
Through to how they get in, transmit, adhere, invade using amazing tricks like actin tails, and then defend themselves with things like T6SS, biofilms, and immune evasion.
and finally, the damage they inflict using those pathogenicity islands and a whole arsenal of exotoxins, endotoxin triggering septic shock, and even mycotoxins.
It really paints a picture of this intense molecular arms race between microbes and their hosts. And it circles back nicely to where we started, with autophagy. Remember how sophisticated pathogens like M. tuberculosis or HSV-1 deliberately block that cellular cleanup?
Yeah, disabling the garbage disposal to hoard nutrients and hide.
Exactly. So, here's a thought to leave you with. If these highly successful pathogens have evolved specifically to prevent cellular self-cleaning because they rely on that messy internal environment,
Hmm.
what if we could develop drugs that force autophagy to happen? Could we effectively make infected cells eat the invaders inside them, turning the host's own recycling system into a weapon against infection?
Forcing the cell to clean house, whether the pathogen likes it or not, that's a really fascinating therapeutic angle. Lots to think about there. Thank you for that deep dive.
My pleasure. It's a complex but crucial area of microbiology.